Good afternoon, everyone. I'm Allison McKay, Chair of the Civil, Environmental, and Geodetic Engineering Department. It's my pleasure to welcome you to this year's season of CEGE CONNEXT Lunchtime Seminar Series. There's a lot of amazing creative research that's happening in the department right now, and the full realization of both the value and the impact of that work really occurs when it's informing practice and policy. This series is an opportunity for you to hear from faculty as they share their perspective on recent innovations that are relevant to practitioners. So we're very happy to connect with you all today and to highlight that the Civil, Environmental, and Geodetic Engineering Department continues to be a resource for you even after you graduate. Today, I introduce Dr. Karen Dannmiller. She is an associate professor and College of Engineering Innovation Scholar. Her research sits at the intersection of building design, air quality, and human health. She's been recognized with the National Science Foundation Career Award, and she is the college's inaugural Be Well Leadership Innovation Awardee. I ask that everyone please mute their microphones during the presentation and drop your questions in the chat function at any time. I will moderate those in the Q&A session at the end of Dr. Dannmiller's presentation. So let us now welcome Dr. Dannmiller.
Thank you so much for being here today, everybody. Thank you so much for hosting this. I'm really excited to talk with you all. So let me share my screen. So today I'm going to talk about the research in my indoor environmental quality research group. So I am someone who never really liked to color within the lines. My research is very interdisciplinary. So I'm in the environmental engineering program here, but I actually have another appointment in public health as well. I am 70% in engineering, 30% in public health, and 0% in microbiology, which basically means microbiology doesn't pay me, but I get to hang out sometimes. So that's fun. But my work is really focused on problems related to our indoor spaces, and especially to the indoor microbiome. So today I'm going to talk about a lot of the work we're doing to look at better ways to prevent mold growth in homes, how to design healthy spacecraft, and also a new way to monitor for COVID-19 using environmental surveillance. So at the beginning of this slide, I like to include a picture of the International Space Station because some of the work I'm going to talk about is going to be from dust collected up there. And I like to remind everybody that you can actually look up in the night sky and see the International Space Station pass overhead, which is really fun and exciting if you haven't done it yet. There's a really great viewing opportunity this Friday, actually. So set your phone alarm right now if you can, 8:57 p.m. on Friday. You can look up some more of the details on that at spotthestation.nasa.gov, but it's really exciting to kind of see it actually travel overhead. So as I mentioned, I direct the Indoor Environmental Quality Research Group. So this is kind of how I think about our indoor spaces. We have our indoor environment where we've made different choices, and those choices can impact the chemicals in our indoor space, the microbes in our indoor space. Those can interact with each other, and then ultimately those might actually impact human exposure, and ultimately human health. So on the engineering side of what I do, I sort of look at fundamentally what's going on in those indoor spaces. How do these different environmental conditions impact the chemicals, the microbes? How are they interacting with each other? And on the public health side, I also look at how that ultimately impacts exposure, especially related to childhood asthma as well. And I like to think about what it actually means to have a healthy indoor microbiome. So ultimately, we have all of these design choices, the flooring material you've selected, the conditions in those indoor environments, our behavioral choices, and how those actually impact our microbiome and what it actually means to have a healthy indoor microbiome in your indoor spaces. So a lot of times people will actually think about, so for the microbes, okay, well, they must all actually be bad. Well, when I think about the microbes in our indoor spaces, I like to sort of think about them in two different ways. And you can kind of broadly imagine these as like the good guys hypothesis and the bad guys hypothesis. So the first is the hygiene hypothesis, which you may have heard of, that we need some exposure to the right microbes early in life to help with immune system development and help keep us healthy. On the other side is the bad guys hypothesis, which is basically that you have some detrimental microbes that are harmful, And if you're exposed to those, you can have harmful health effects. And so you might think of this as mold growth or exposure to pathogens and things like that. In reality, the vast majority of the microbes in our indoor spaces are neither of these. We're really fairly indifferent to them being there at all, but we can learn a lot by studying those microbes that are potentially beneficial and are potentially harmful. Today, I'm going to focus mostly on the bad guys hypothesis. I do have some work on like looking at childhood asthma development, on potentially looking at the good guys, but we're going to focus on the bad guys today. So when we think about these bad guys, today, exposure to mold in housing actually costs about $22.4 billion a year. That's specifically exposure to asthmatics in housing. And it usually doesn't look as pretty as this piece of artwork over here. It makes up about 16% of the costs associated with allergic rhinitis, acute bronchitis, as well as asthma. And so it's actually a big problem. And we've even seen this in the news recently with all the really unfortunate flooding events that have occurred. There's going to be a lot of mold that impacts the people who are affected by those. And it's really an unfortunate situation, really a growing problem. So mold growth in housing is harmful. It's harmful on Earth, but it's also harmful in space. And historically, space stations have actually had problems with excessive moisture. So if you think back to Mir, the space station Mir, at the time of its decommissioning, it actually had fairly extensive fungal growth occurring. The fungal growth was occurring on things like wiring and window seals, which obviously you really want when you're in space to make sure that those remain intact. We learned a lot of lessons when we built the International Space Station, and we were able to much better control the relative humidity up there. But you still have this completely enclosed space with no ventilation, with people exhaling moisture, breathing, sweating, putting moisture into the air. And you can still have unintended situations, such as this is an example of a panel where the astronauts were hanging their damp towels in the hygiene area, and there was mold growth that then occurred on the panel, and it had to be removed and thrown away. So not only is a problem here on Earth, but also a problem in space. So a lot of my talk today is going to focus on the mold growth, both in our homes as well as in space. But I did also want to spend some time talking about the pandemic and some of the new viral monitoring techniques that we've been developing, thinking about the indoor microbiome. So when we think about the indoor spaces, we can actually think about how using the information we can collect even from dust in our buildings can actually help us monitor what's going on in terms of our viral exposure. So overall, my talk is divided into 3 parts. The first is everybody's favorite topic, moisture and carpet. At least it's my favorite topic. Maybe it's just me. And then part 2 is spacecraft. So we're going to talk about some of those potential unintended consequences if they could occur through like a ventilation system failure or something like that in space. And then finally, we'll talk a little bit about what's been on everybody's mind for the past 3+ years, which is infectious disease and COVID-19. So let's jump in on moisture and carpet. So previously, when people think about mold growth in housing, a lot of times they think about mold growth on drywall. So you think about the picture I showed earlier, you have maybe damp conditions in your home, you have this like visible mold growth on drywall or like in your bathroom, somewhere like that. But we wanted to ask the question, could you also potentially have fungal growth in carpet? And we wanted to focus on potential fungal growth in carpet for a couple specific reasons. The first is that carpet is actually very prevalent as a building material. It currently makes up about half of the US flooring market and is really prevalent post-World War II. So this shows that about half the homes surveyed in this, it was actually for a magazine, had carpet, and that's still approximately true today. In addition to being prevalent, it's also a really important source of human exposure. So we actually held a workshop entirely on carpet here at Ohio State. We held it in 2019. We almost held it in 2020. Thank goodness we did it in 2019. But we invited 40 experts on carpet to come and talk about what it means to have a carpet in your home. And this included people from the carpet manufacturing industry, from the carpet cleaning industry, from all different areas of academia, engineering, public health, etc., as well as government agencies and nonprofits, and just ask the question, what does it mean to put a carpet in your home? And one of the big conclusions from this workshop was that a carpet is actually a really important reservoir. So that means it's actually a source and a sink. When you have dust in your indoor space, it can actually end up in the carpet, kind of embedded there. And when someone walks across the room, they resuspend it out of the carpet more so than they would from a solid surface flooring material. And that can ultimately lead to human exposure. So it's this really important source of exposure to dust in our indoor spaces as well. Finally, the last reason that we wanted to focus on carpet is that the carpets can actually hang on to moisture in the indoor spaces. And so this graph shows the relative humidity in the carpet relative to the room air. And you can see that it is able to sort of hang on to that moisture longer than you would have in, say, the center of a room. And when these materials get damp, it can have a lot of impact on things like dust mites and the chemistry that might be occurring, as well as bacteria. But we really wanted to focus on the potential for fungal growth that would occur in the carpet. So what we did is we actually went around Ohio and we collected matching carpet and dust from homes. So this is our before and after pictures where we cut out a rectangle from the middle of the room. All of these carpets were people who were getting rid of the carpet for aesthetic purposes. Maybe they didn't like that pink, purple, polka dot, whatever carpet and wanted a new color. I like to say that we're going to start a renovation company. So if you want a rectangle cut out of your carpet, just let us know. And we collected the match dust samples from those homes, brought it back to the lab, and then incubated the carpet under different relative humidity and temperature conditions and looked at microbial growth rates, function, as well as other impacts on indoor chemistry as well. So when we bring the carpet back to the lab, we embed the dust in it. We embedded the dust using this modified ASTM method. ASTM actually has a method for everything. If you ever want to put the dust back into your carpet, highly recommend. This is very effective. You just roll a heavy pipe over it and then put it into these temperature relative humidity controlled chambers and looked at the microbial growth that occurred under different conditions. We use a lot of next generation sequencing technologies and other DNA-based techniques to look at the microbes in these samples. So we'll take the dust out of the carpet, extract the DNA, and then we can actually get identification of what microbes and how much are present in all these samples under the different moisture conditions. So the first thing that we noticed when we did this is that we actually saw visible differences in the dust when we incubated under these relative humidity conditions. So when we measure the moisture in the dust, we actually use a measure called water activity. It's different than water content because it's a measure of water availability to the microbes. So it's often used in the food industry. So if someone wants to know if your granola bars are going to, grow mold on them, they can actually use water activity to measure how much the water is available to those microbes. Takes into account things like salts and sugars that might bind up some of the water, similar to how you couldn't drink a cup of ocean water. And when you actually visibly look at the dust, you can actually see visible differences. And we knew we were onto something because at 100% relative humidity after 1 week, we actually saw visible fungal growth. And the water activity equilibrates to the relative humidity pretty quickly. You can think of it like a equilibrium relative humidity in our jar at like 85% is equivalent to a water activity of 0.85. So they're sort of equivalent like that. And relatively quickly, we saw after 1 week, incubations at these different relative humidity conditions that you do get fairly extensive fungal growth. Our original dust sample here, you can see that at 100% relative humidity, you actually get 27.5 times as much fungal growth as was originally present in the dust. You don't get as much bacteria except at 100% relative humidity. You compare the bacterial concentration to your relative humidity levels. It's really just at 100% that the bacteria shows up. So it's really all about the fungal growth or the mold growth in these dust samples. That growth continues over time. If you look at the bottom two graphs, so you can see we did 100% relative humidity and 85% relative humidity. We were going to do 100% relative humidity out to 6 weeks, but the carpet samples were so extensively covered in fungal growth, we figured we had kind of made our point by that point. But we were actually able to show that microbial growth does occur in carpet and dust samples as the relative humidity gets above about 80%. And this was also important because the moisture is really the limiting factor here. We measure the nutrients that are needed to support this growth in dust, and none of these were limiting to actually support really extensive growth over the course of the week. The nitrogen was the only thing that was even close, and it still is not limiting. So really all of our indoor spaces, and our dust in particular, is just sitting there waiting for moisture, and it's really the only ingredient that it needs to help that fungal growth start occurring. Excuse me. And so the growth in the dust also potentially contributes to our aerosol exposure. So this is a rough aerosol resuspension model, and you can see the amount of exposure you'd have to fungi and bacteria in the orange, and then the additional exposure you get after these elevated relative humidity conditions for a week. And you can see that you do get an additional contribution to your aerosol exposure in these rooms, mostly for the fungal exposure. And this is especially important because the species that tend to grow here are the ones that tend to be the ones that contain allergens that you might be especially concerned about for exposure. So the next study that we did, we wanted to extend our original work out to some additional homes. So we collected more homes, 19 homes from the metropolitan areas in Ohio. And this is the work from Sarah Haynes, who's now a assistant professor at University of Toronto. And we collected some additional samples, and we wanted to look now at variations in relative humidity. So nobody leaves their house at exactly 90% relative humidity for a week, or at least I hope you don't. But more realistically, what tends to happen in homes is you have a short burst of elevated relative humidity. Somebody takes a shower, somebody has a cooking event where a lot of moisture is released into the indoor space, somebody opens a window during a hot, humid summer day, something like that. And so we wanted to ask the question, if you have shorter periods of elevated relative humidity, can that still support fungal growth in carpet that might be important for human exposure? So we collected the same samples, and this time we incubated them under varying relative humidity conditions. And so you can see we elevated them. They were at high relative humidity for 0, 6, 8, 12, 18, or 24 hours per day. And we asked if we could model this growth using the time of wetness framework. And I will save the mathematical details of this for hours 3 and 4 of my talk. But you can basically think of this as the percent of time that the sample is wet, the time of wetness at the bottom, is proportional to the relative growth rate, or how fast it grows. So the more time that it's above this like critical growth threshold that it needs for moisture, the faster it's going to grow. And there's 4 different models here. The standard model sort of has this one-to-one association, but there's 3 other frameworks within this that you could think about potentially using to model the growth that you might get under these different, more realistic scenarios. So when we looked at the data, we were excited to see that we could actually indeed use this framework to model this growth. So in this case, if your maximum relative humidity is either 85% or 100%, it looks like what most closely fits our data is this 2 activation limited growth model, but we need a couple more data points down the road to prove that. But even the main takeaway from this is really that even short bursts at elevated relative humidity are sufficient to support fungal growth in carpet that might be important for human exposure. This is especially important too, because when you have these shorter periods of time with elevated relative humidity conditions, if someone takes a 20-minute shower, for instance, the relative humidity on the wall tends to stay elevated above this 80% relative humidity growth threshold for about 6 hours. So this really can support this fungal growth, and explains why in so many bathrooms we do start to see this fungal growth occurring. The US EPA recommends that the relative humidity in your home be between about 30 and 50%, and definitely below 60%. And that's a pretty good recommendation because it does keep you below that critical growth threshold and far enough away from it that you can help prevent this growth from occurring. All right, so I'm going to show you another graph, and this graph is a mess, and I'm showing it to you because it's a mess, but let me explain why. So this is a principal coordinate graph where similar microbial communities cluster together. So if 2 dots are really close, the microbes in those samples were really similar. If 2 dots are really far apart, the microbes in those samples were really, really different. And this is all of our 19 sites from Ohio, colored by site. And then the darker is the samples that were exposed to higher moisture conditions. The lighter were exposed to lower moisture conditions. And what we were hoping to see here is a moisture signature in the microbes. So we were hoping for one clump of samples all together that was really dark and said these are the microbes that are found when it's really damp, and another clump of samples that were really light, and these were the microbes that are there when there's no dampness. And that's not at all what we saw. As I said, this graph is a mess. And in fact, we didn't get samples that clustered that way. They cluster more strongly by site. So you can kind of see here's a blue cluster, here's another different blue cluster, here's some like pink, purple, pink, green is down here. And there's really not a consistent microbial community that shows up when moisture occurs. And so what this tells us is that we really need to be thinking beyond what species are present to determine if a house has dampness. So a lot of the different methods we have to measure mold in housing are based on sort of who's there, what microbes are present. And this says this may not be necessarily the best way to actually do that, because each house is so unique. It has such a unique unique microbial signature that moisture doesn't change it enough. Each space is just so unique in and of itself. So to go beyond this, we wanted to look now at more microbial function. So if it's not who's there, let's look at what the microbes are doing. So this is the work in collaboration with Bridget Haggerty at Case Western Reserve University. And what we wanted to look here was what is actually happening, what the microbes are doing. So now we're looking at RNA, which basically is the gene expression. What are the microbes doing under these damp conditions? And so we incubated some of our samples at 50%, 85%, and 100% relative humidity. And when we look at the function, the 85 and the 50% are pretty similar to each other, but all of a sudden at 100%, you get all of this different microbial function. And not only are they different from what happens at the lower levels, but they're very different from each other. And you get all this metabolic diversity. This is a GIF from the work. It's my favorite GIF. I'm still waiting for it to go viral on social media. So look for it on your feed. But we looked at the metabolic pathways that are happening in the fungi in these dust samples at 50%, 85%, and 100% relative humidity. And what you'll notice here is that at 50%, it's really the core metabolic functions that are happening. Lipid metabolism is complete, carbohydrate metabolism is complete, but those fungal cells are really just hanging on for life. There's a little more at 85%, but at 100%, you suddenly get all of the outside, the secondary metabolic pathways lighting up around the outside of this graph. And that's really important because that tends to be where the allergens are, and the mycotoxins, and the virulence factors, and all those things that you're really interested in terms of human health effects that really light up when the microbial function changes around the outside of this graph. So we wanted to ask, is this true? Do you get more allergens per spore if you incubate these at higher relative humidity conditions? And indeed you do. So actually this graph on the left, I won't go through the details here, but we do see statistically significant upregulation in allergens for samples incubated at higher moisture conditions. And we also looked at a protein-based assay for one species here, and we actually saw higher reaction in human serum per spore for spores incubated at higher moisture conditions. So this really begs the question of, is a spore, or does it matter how it was grown? Is it really all about that moisture? And are these higher moisture conditions setting up the stage for these increased health effects? We also looked at some of this data. So some of this is work by Nick Nastasi, who's a senior PhD student in my group. And we looked at other things that potentially matter. So we focused here on one specific allergen, ALTS-A1, which is a fungal allergen. And we also saw that allergen specifically was more upregulated at higher moisture conditions. In this study, I'm not going to go through all of the details here, but we also looked at what factors might play into how much fungal growth you get. Moisture is by far the #1 factor in terms of, again, it's the limiting factor in terms of this fungal growth that you get in carpet. The amount of dust does play a role, not as much as moisture. And for most normal carpet loadings, it's actually not really going to matter. The amount of dust only really matters if you have like a brand new carpet that somebody just put down and hasn't really gotten dirty yet. And then finally, the carpet fiber type does matter, but not as much as the dust does either. So if you do have different fiber types, that can influence potentially how much growth you get, but really it's all about the moisture and about the dust, which are a little bit more important. So these next images, apologies, you can't unsee these, but these are some SEM images we took of the fungal growth in carpet. So the larger fibers here are nylon carpet fibers. And this looks pretty similar to what we've been seeing in a lot of our other measurements. At 50% relative humidity, you don't get a lot of fungal growth. You can just see there's some dust particles in our carpet fibers. At 85 and 100%, you start to get hyphal extension. And then finally at 95 and 100% relative humidity, you have extensive fungal growth, presence of fungal fruiting bodies, and things like that. So this just looks like kind of what we had been seeing before. We also took a confocal Z-stat because we wanted to kind of understand what this looks like in 3 dimensions. So the nylon carpet fiber here is gray, and you can see some of the fungal growth wrapping around that fiber in blue as well in terms of what it actually looks like. So overall for part 1, we saw that elevated moisture in an indoor environment is really sufficient to support fungal growth and function in dust and carpet. Even if you have short bursts of elevated relative humidity that happen consistently, that's sufficient to support that growth, and it does change the fungal function in ways that might be important for human health. This has a lot of important implications for health as well as building design in terms of thinking about controlling moisture in our indoor spaces, and how we want to try to prevent those different exposures from occurring as well. And I do think the US EPA recommendations about controlling relative humidity, definitely between 30 and 50% and below 60%, is generally a good recommendation to help make sure that you don't get that unexpected growth. All right. So now I'm going to move on to part 2. So let's talk about this now in outer space. So we talked already a little bit about what this potentially looks like in our indoor spaces here on Earth, but we want to ask the question, does the same thing happen if you now expose these microbes to moisture in dust from the International Space Station? So this was a collaboration done with NASA, where we did get dust down from the International Space Station. So every Saturday, the astronauts up there will actually go and vacuum the HEPA filters that filter the air in the International Space Station. You can find some really cool videos of it on YouTube if you take a look. And they collect it in a vacuum bag, and a lot of times they would actually just throw that away to burn up in the atmosphere. And we were like, no, please send it to us. We would love to have that. So they did bring some down for us. It came in this really awesome box that says critical space item. Very, very cool. And we were able to take that dust and then incubate it under these conditions to mimic what might happen if you had, say, an unexpected ventilation system failure or other unexpected high moisture event occurring. So you could actually visually see in the dust the difference between incubation at 50% and 100% relative humidity, that you get visibly darker dust occurring here. And one thing you'll notice about the dust, it's very linty. The dust from the space station, it's probably from the astronauts' clothes, them moving around up there. And it's very similar to working with carpet, actually. So as we were working with the dust, we were just thinking about how really we've been training for this for many years in our work with carpet to handle the dust from the International Space Station. So indeed, having a higher relative humidity on the bottom does increase the fungal concentration in the dust, shown on the x-axis after incubation. In this case, we incubated for 2 weeks at all of these relative humidity conditions. And so this is actually very similar to what happens on Earth. It's really just that moisture is a limiting factor as well. It's just, all the ingredients are necessary there, except the moisture. So it does emphasize the importance of controlling the moisture in these other indoor spaces too. Both the length of time and the actual relative humidity itself help determine what growth occurs. And I won't go through the specifics here, but basically it looks like you have a similar 80% growth threshold, some of the other data we have as well. and that it's at these higher relative humidity conditions that the growth occurs pretty rapidly. We did also apply the time of wetness framework here to see if we could actually calculate how much growth you'll get under different moisture conditions, even if the relative humidity is fluctuating over time. And it was really interesting because it looks fairly similar to it on Earth, where you have this activation limited model or the 2 activation limited model, except at 100% relative humidity, we got a slightly different framework, the deactivation limited model, from the space station dust. So we're not sure why, but it's really interesting and something that we might be able to look into a little bit more. We of course took some pictures. So the original dust actually looks great, looks really clean, similar to carpet fibers on Earth, as I mentioned. These are really similar materials. We don't see a lot of evidence of problems or anything from the original dust itself, but if you incubate it at these higher moisture conditions, you again get this fungal growth occurring, which again models sort of this ventilation system failure scenario. So this was really helpful to NASA. We could provide to them some information and recommendations about when they might expect to have a problem. If there is an issue up there where the relative humidity gets too high, when can they anticipate that there is a problem that they need to take some steps to remediate it and clean it up? And of course, it also helps to know that the dust up there is actually really similar to the dust down here. That if you look at these in parallel, the International Space Station dust as well as the dust from homes on Earth, there's a whole lot of parallels and actually behaves really, really similarly with just some subtle differences too. So take a look, International Space Station, 8:57 p.m. on Friday, and you can now think about the dust up there as well. So overall, the microbial growth on spacecraft is actually really similar to the microbial growth in environments on Earth, with some just very slight differences. But we were able to help develop this model to provide this information about when you might potentially expect a problem from elevated moisture conditions on spacecraft. All right, so I am now going to shift gears a little bit. So we've been talking all about fungal growth, and I said, at the beginning we were going to talk about microbial good guys and bad guys. This is a bad guys talk. So we are going to talk again about another bad guy, the SARS-CoV-2 virus and COVID-19. And so when the pandemic started, as the Indoor Environmental Quality Research Group, we were sitting there and thinking, how can we possibly contribute to what is going on in the efforts that are underway to potentially help fight against COVID-19? And we think a lot about buildings. And so we wanted to see if we could develop a new long-term surveillance solution that could be used at the building scale. So you may be familiar with wastewater monitoring, which is really effective and a great way to monitor for COVID-19 outbreak, especially at the entire sewer shed level. And it has a lower monitoring cost and a lower effort, and it helps you monitor a large population, but it doesn't provide as high resolution data. On the other end of this is individual testing, which is everybody's favorite thing to go get tested, right? Not really. But it has a higher monitoring cost and effort, and you're monitoring just whoever you're testing at that particular point in time. We wanted to see if we could develop a kind of mid-range option where you could maybe look just at buildings that are of particular interest or concern. And this is especially helpful because it can be challenging sometimes to do wastewater monitoring at the building scale. Not everybody sheds virus in feces. Depending on the building type, people may or may not use the restroom there. This is a respiratory virus, of course, and not all buildings actually have an appropriate access point. Sometimes you get a point where you're monitoring a lot of buildings. So we wanted to see, is there another way that we could do environmental surveillance for viruses at the building scale? So the first thing we started to look at is thinking about a sneeze, right? Everybody loves this picture. And we think about, where does the virus go? So if you think about these droplets, the transmission occurs a lot because of the really tiny droplets. They get in the air, they stay in the air for a really long time. Where do the large droplets go? Well, there's a lot of large droplets, and those also fall immediately to the floor. The viruses in these droplets are actually present in a range of particle sizes. So you can think about a VOC as like the tiniest dot here, something that you smell. The virus is sort of in the middle, but it's usually not floating around on its own. It's generally in respiratory droplets from breathing, coughing, sneezing, talking. And those tend to be in the range of 0.1 to 100 microns in size. And if you think about where those particles end up, on this graph, you'll note that this is on a log scale. You have particle diameter on the x-axis and deposition velocity, or how fast something settles out, on the y-axis. And you'll note that really small particles are removed quickly from the air. They're actually really sticky. They're removed by electrostatic forces of Brownian motion, and they tend to stick to like walls and ceilings and things like that. The larger particles tend to settle via gravity, and they fall quickly to the floor. It's these particles that are right around 1 micron in diameter that settle out really slowly, and they tend to stay in the air for a really long time. And those are the ones we're especially concerned about relative to transmission. But a lot of these respiratory droplets fall in this larger size range and sort of end up settling out to the floor. So viral transmission actually happens via multiple routes. I'm not going to go through the details of this, but here we're not actually interested in transmission. We're interested in where the virus actually ends up in indoor spaces. And if you think back to the carpet workshop we remember again that dust serves as this microbial reservoir. It's this basically memory of what happened in those indoor spaces. And people previously had shown that other viruses were present in floor dust. And so we wanted to ask the question, basically, could dust potentially be a way to monitor for COVID-19 at the building scale? And think about developing this sort of long-term surveillance solution at the building scale using potentially building dust. So what we did is we started by collecting samples from rooms of students in isolation for COVID-19. This was in the fall of 2020. And so these were rooms where everybody had tested positive for COVID and they were waiting out their isolation period. And so we collected bulk dust from these rooms, as well as surface swabs and a passive air sampler. In these samples, we measured viral RNA. So if you think about what the virus looks like, it looks kind of like this. It has an envelope on the outside. It has this capsid in the middle, and then the nucleic acid or the RNA in the center. When we're measuring this virus, we are just measuring this RNA in the center. The envelope on the outside is really fragile. It tends to break down in these indoor spaces. So basically, it tells us nothing about infectivity. The envelope is necessary for infection, but we are only measuring the nucleic acid in the middle, and not the envelope on the outside necessary for infection. So what did we find? Well, when we looked at concentrations, we actually found that the highest concentration was in the floor dust from all of our surface swabs as well as our air samples. And we also found the highest percent positivity in the floor dust as well. So our air samples were only 20 or 30% positive. And remember, these are rooms where everybody has COVID and is recovering. Our surface swabs were 55% positive, but our bulk dust was on the first pass 89% positive. And with some improvements in analytical techniques, we quickly got that up to 97%. And now we've been doing this for years, and we're probably somewhere closer to 100 even in terms of that bulk dust positivity. I mean, so really the answer here of like where you want to measure potential viral deposition is actually in the dust. Interestingly, the RNA in that in the dust remains pretty consistent over time once you've collected the vacuum bag. So on these graphs, you can actually see as we were measuring it over time, and we didn't see really decay out to 4 weeks as we did in this study. And it does decay in the real buildings, probably through other processes like dust removal and cleaning. But if you collect a vacuum bag, and what we can think is probably that protein capsid right around the RNA holds that RNA there stably. So you don't need to store the dust in a cold storage. You can actually just use it for the vacuum bags after you collect it. This was also despite the application of a disinfectant in this room prior to sample collection. So prior to sample collection, because of restrictions at the time, this disinfectant was sprayed around the room and let sit for 20 minutes before the dust samples was collected. And even with application of this disinfectant, we were still able to pick up RNA from the dust samples. So based on this, we were able to also do a small pilot study starting in the spring of 2021. And this is just from one of the residence halls, also on a log scale on the y-axis, you'll note. And we collected samples from both the custodial staff that had collected dust samples from the main common areas, as well as the private student vacuum. Then this was a vacuum in a closet that students could like optionally use as they wanted. And we saw pretty good correlation here with actually the percent positivity in this building at the time relative to the concentrations that we were picking up from the dust samples. So that was encouraging. And we also noticed that we could even sequence the variants out of the dust samples and track that over time as well. So that was really exciting as well. We were able to then scale this up. And so we've been sampling about 50 buildings a week since August of 2021. And you can see this is data from fall of 2021. And it corresponded really well, our data from the residence halls and the buildings on campus relative to known outbreaks in the population at the time. You'll see some of our early data in this semester. We don't see a lot of higher levels. There was an outbreak at that time, but interestingly, people had to test before entering the residence halls. And it looks like we were able actually to catch a lot of those individuals before they entered the residence hall, so we didn't see them in the dust data. We do see kind of the end of that outbreak earlier, then there's a lull, and then we had another outbreak corresponding to the winter that we were able to pick up in the dust data as well. This data is a little complicated because the viral shedding does occur over time. And so if you think about what's happening in these buildings, we have somebody becomes infected. At some point, they get a positive test, then they were moved to the isolation room. Then they come back, they might still be shedding some virus as they return, at least the RNA, even if they're no longer infectious. And so this data is complicated. We're working with some biostatisticians now to figure out the best way to process this data and figure it out. But overall, it looks like this is a pretty good strategy to actually monitor for these viral outbreaks. I also wanted to highlight some work by John Van Dusen, who's a PhD student in my group, and he is also looking at the sequencing the variants out of these samples. We were actually able to pick up the transition from Delta to Omicron that occurred over this period of time. So you can see it kind of happened over the holidays in early 2022. And in our dust samples, you can actually see that transition occurring. This is just from one particular building, one of the residence halls, and then this is from a multi-story building where we actually had some early samples with the Alpha variant. You see the Delta variant in the middle, and then that transition to Omicron that occurred over the holidays. And we actually have this data across campus that corresponded very well to the individual data that we had for testing what variants were emerging. So I like to emphasize again, what does this mean about viability and dust? Absolutely nothing. So I just want to emphasize again that here we are measuring the viral RNA in the center of that virus. It doesn't tell us anything about that viral envelope. So we did want to try to understand, though, a little bit about what that might potentially look like. So we did do a study looking at viral decay on carpet and dust. And so this used two viral surrogates. So these are bacteriophages. That means they are viruses that infect bacteria only. They can't infect humans, and so we were able to use them in our lab safely. And we applied them to carpet and dust. And MS2 is a non-envelope virus, so it's missing that envelope on the outside that's really fragile. And it's similar to something like norovirus. Vi6 is a bacteriophage that's more similar to SARS-CoV-2, the virus that causes COVID-19, and it has that fragile envelope on the outside. And what we saw was really interesting, that actually the envelope virus decays very rapidly on carpet and dust samples compared to the non-envelope virus, which can persist. And this is very similar to other surfaces as well. We see in the literature that a lot of the non-envelope viruses can persist for a relatively long time, where those with envelopes like SARS-CoV-2 tend to decay rapidly in terms of infectivity in the environment. However, we see again the RNA that we can detect persists for a really long time. And so this was just a shorter time, but we've seen in other studies the RNA can persist for a really long time, even if the virus is no longer infective. So as we get these environmental measurements, they actually tell us nothing about the infectivity of these viruses. We also looked at removal by cleaning methods for both of these bacteriophages for MS2 and 5/6. And we saw that different cleaning methods were a little bit more or less effective. So concentration on the y-axis and different cleaning methods on the x-axis. And we actually saw that the envelope viruses were a little bit more efficiently removed by these cleaning methods. But vacuuming and hot water extraction didn't do a great job of removing the non-envelope viruses. Here, when I say hot water extraction, it's actually what most people think of as steam cleaning. It's where you apply hot water and then suck it back up. We did actually, for this study, apply actual real steam as well, which is not typically done in real indoor spaces. But the steam and the application of the disinfectant were fairly effective at removing the virus. And the RNA of both these viruses here in Part B did tend to hang around even despite a lot of these cleaning methods. So overall, we saw that dust can complement the wastewater monitoring pretty well. So in terms of wastewater monitoring, you can monitor a much larger population or potentially small population. But in some cases, the dust can really step in to help with this data. For wastewater monitoring, the sample collection can be really difficult, requires these intensive pre-concentration steps, and not everybody sheds virus in feces. So really, the dust method here can be a really nice complement to other monitoring efforts that are going on at different points in time. And it's another tool in the toolbox that we can use to understand what's going on in these different populations, especially also that can be used to target vulnerable buildings that contain vulnerable populations where we might really need to know what's going on. And we might also need to know what variants are potentially circulating in those areas. So we've been, continuing to monitor this and help provide this data and information about what's going on. So overall today, I talked about 3 different things. So first I talked about fungal growth in indoor spaces and dust and carpet. And we showed that elevated moisture is really sufficient to support that microbial growth and function in dust. We also saw that was true on Earth as well as in space. So this applied to data from the International Space Station as well, modeling something like a ventilation system failure. And finally, we talked about another bad guy, SARS-CoV-2, and how dust is a potentially efficient matrix to monitor for COVID-19 and understand basically what's going on in the population and be able to track that as well as the variants that are present. So I have a lot of people to thank for this work. Of course, all the funding agencies that funded this, as well as some awesome collaborators. And of course, the fantastic students who they're the ones are in the lab every day working hard to get this data. So it is a jungle out there. Thank you everybody for your attention. I'd love to answer some questions.
Oops. Okay, actually there is a question as to whether there's any opportunities for industry to consider different materials or designs in order to help with processes around indoor microbial quality.
That is an excellent question. So with the workshop that we had in 2019 at the carpet workshop, it was actually really great to meet with some of the carpet manufacturers. A lot of the carpet around the country is manufactured in Dalton, Georgia, and the history goes back to this one woman who sort of like a long time ago really perfected the art of making similar textiles that then evolved into carpet as we know it today. And so all the manufacturers are still located in basically that area. And when I talk to them, they have a lot of capabilities, of designing materials in different ways that could potentially help do things like reduce resuspension, or things like that. And some of it does come a little bit down to what I believe is a little bit of an education piece that people should be interested in that, because right now they don't focus on it as much. So I think some of it's getting the word out, but they still could potentially market it in that way. And I think potentially working in industry to focus on those things and manufacturing carpets in that way would be beneficial to indoor environment, indoor environmental health and things like that. Some of the coatings that end up on carpets can potentially be harmful. So if you do things like put an antimicrobial coating on there, you also have to worry about exposure, especially to children, of those coatings. You don't want to be doing things like putting PFAS on carpets, or things that can potentially result in high exposure, because you think about the littlest ones are crawling around on those carpets, putting everything in their mouth, and are going to ultimately end up with exposure to those coatings. So it is better if we could stay away from those coatings. The industry as a whole has moved more towards polyester materials in carpet. So as we did our study, we focused more on like older carpet materials. And then since then they've kind of transitioned more to polyester. So we were like nylon, olefin. Now most of the carpet market is polyester materials, which has some benefits of being stain releasing without the use of coatings, but some of the carpets still have those different coating materials on them. So I do think, though, I would like to see more of a shift towards thinking about some of these different properties as people manufacture carpets as well. A lot of people think that I'm anti-carpet, and that's actually not true. There are benefits to carpet in our indoor spaces in terms of injury prevention, sound dampening, just feeling comfortable, things like that. But I do think we have to think carefully about where we put a carpet. I don't think we should be putting carpets, wall-to-wall carpets in bathrooms, on concrete floors in basements where they're going to get damp, things like that. And I do think I would like to see a little bit more focus on some of these environmental health concerns in terms of reducing resuspension and how we design them. So it was kind of a long answer, but yes.
Okay, any other questions? All right, well, I want to thank everybody again for joining us. And we'll have the CPD hour credits out in about 2 weeks for those of you who had requested that. And our next session will be in November, and we look forward to having you join us at that time. Thank you.
Thank you.